Good afternoon, everyone. This is another Wolfram Institute live stream. Today we have Nicholas Frieler, and he'll be talking about analyzing multi-way chemical pathways using chemical space networks. All right, take it away, Nicholas.
All right, thank you. All right, so to start off, we're going to be talking about just um, sort of, I guess we'll define the terms first. So a chemical reaction network is something like you see in the image here, where you start with one of the molecules, and then in this directed graph, an arrow indicates that some sort of reaction you specified takes place. So if we go down, there's just some setup stuff here. So here is a condensation reaction specified in this way. And basically you have these two components, which is just any sort of, the star indicates that it can be like any sort of element. It's not specified here. And then there's this OH group. So you have two things of this sort. And then a condensation reaction combines them together in this way. And then you have a water molecule left over. So this is just pattern reactions in the Wolfram language. And then if we want to sort of utilize the functionality of a multi-way network, what we can do is we can start with a bunch of these molecules and have a set of reactions and apply every possible reaction at every possible place on the molecules and sort of build up our multi-way network from there. And depending upon the reactions you specify, you can get just a few products. So here we have a condensation reaction that specifies that it has to be like attached to a carbon. And then this general one is it can be either carbon or nitrogen. And you can see if you start with just a few molecules, you can get pretty reasonable sets of output molecules, right? So this graph isn't going to grow as big because you don't have that many reactions. But if we scroll down and work our way through this, um, you'll see here we can get a lot of reactants after just two reaction steps, right? So if you start off with this one, the reaction we're doing here is this sort of reaction here, where you combine a diene and a dienophile and get a product that looks somewhat like this. So starting with this, there's only three possible products, but if you apply that reaction step again, you get this set of 11 products. And then as you continue to let this happen, you can get like more and more, and it grows exponentially, the number of products you can get, because this reaction is we haven't imposed very many restrictions on this. We haven't said that it can only form molecules of a certain size, and we're not in the way this is written, we're not necessarily like conserving mass, which may sound a little confusing, but in the way pattern reaction is set up, it's not like you have three specific molecules with maybe a total of 12 carbons, and then throughout the multi-way graph, those 12 carbons are conserved. What it actually does is it takes one of the molecules in the set you have, and then any other molecule in that set and combines them. So it doesn't conserve mass in that way, but this allows us to see a much larger range of reactants. And if you have a bunch of molecules together in some sort of solution, right, you can't really control for which molecules are interacting with others. And so this method of just saying we'll combine any two things possible works really well for that kind of setup. So that's the multi-way reaction network. And now a chemical space network. Uh, what this does is it takes a large collection of molecules and then tries to group them based off of some criteria you've specified. So the one I did for my project that I did over the summer was I looked at the topological fingerprints of these molecules. So what are the ring sizes? How are the rings next to each other? What various chains are in these molecules? And then try to group those based off of those similarities. And so, and then you can look at various things relating to these. So if you look at a sort of similarity score for the topologies, um, a higher similarity score just means the topologies of the molecules are more similar. And if you group them into connected components based on how similar they are, you can look at things like the number of non-singletons remaining. So this specific one basically means if I say my molecules have to have a similarity of at least 80%, how many molecules left out of my group are no longer that similar to any other molecule. And for 80%, you can see it's about 13 of them are singletons, so they're not similar to any. And obviously, as you increase, you're going to get more that are not similar to any other molecules. And more often than not, depending upon how you generate these molecules, you're going to get, they're all going to be, it's going to be one large connected component in terms of similarity until about like 50 to 70% similarity. And then you're going to have this drop off. And then another thing you can look at is the number of connected components. So as you increase the similarity threshold, how similar the topologies need to be, you're going to get more splitting of these connected components, and you can see how this grows. And they all have roughly similar growth patterns between, depending upon how you generate these molecules. It's generally kind of flat and boring for these lower numbers, and then you get this sort of gradual and then steep increase. And the specifics of it will change with every set, but the general trends are about the same. And then also the size of the largest connected component. Again, it's connected up until about 50%. So it's going to just be all the molecules, and then you're going to get splittings. So one group will break off, then another, and then it'll continue to decrease. Okay, and this is just what some of the similarity networks look like, right? You can see that you have this, it's just one connected component, but it seems that these two groups are not quite as similar to each other. They only have a few nodes connecting them. And once you go from 0.5 to 0.6 similarity threshold, you get a splitting of these two groups. And then as you go, you can see this group is going to split soon. And some of these others are, the connections between the nodes are more sparse. And then once you get high enough, you just get all of these small clusters and some of these singletons here. And
So this
really,
is, you have a set of all these molecules, and you're making connections between them if they have a certain topological up to a threshold, a similarity in topology.
yes, yeah. So there's so the specific topological similarities I looked at was this topological fingerprint of a molecule, which is part of the Wolfram function repository, and essentially it converts every molecule into a about 2048 bit fingerprint based on its topology, and then you can just compare the individual bits of two different fingerprints to get how similar they are.
Okay. Is that agnostic to the actual what each node on the graph is? That is the atoms themselves. Does it care about this one's carbon, this one's nitrogen, that kind of thing?
I believe, from what I recall, I think it does care about that. Because I seem to remember having, one of the things I did look at was also, okay, I can, like, looking at these two molecules, I can tell them apart, but topologically they're listed as identical, and trying to figure out, because it wasn't immediately obvious. It looked like there were topological differences. But I don't ever recall seeing like, oh, this one has a nitrogen and this one doesn't have any. So I think it does care to some extent which elements are in there. I'm not sure if it's, just the total number, or if it takes into account the positions of them, but I think there is an element of that in there. Okay. And so one of the things that's really cool about these multi-way networks and then chemical space networks is we can combine the two in two different ways. So the first way we're going to look at is we're going to use the multi-way network to sort of lay out our various molecules, and then we're going to use the chemical space network to color them and to get different clusters. So what we have here is this, like, the diene and dienophile, this monoterpene reaction, and then so we've carried it out 3 different steps, and you can see this is the one we started with. And so that's how we generate the this group of molecules here, and then we use a chemical space network and the topological fingerprint to group them into clusters. And then, as you can see here, we've imposed a similarity threshold of 0.9 and used that to color them. And so each color represents a different connected component, except for gray. Gray is used to represent the singletons or any clusters that are too small to, to be useful in looking at it as a group. And so what we can see here for the most part is that things that are close to each other in this multi-way network have similar colors. So we can see all the yellows are on the right here. All of these like teals are in the upper right, and then most of like the reds and oranges are in these individual clusters, but there are some scattered around. One of the things that this graph doesn't entirely show is that these reactions were only carried out 3 steps. If we carried it out another step, some of these like different branches would coalesce. So while the distance between 2 molecules on this may look like it's 5 or 6, if we carried it out another reaction, it might only be 1 or 2 apart. So even though it looks like there may be some like breaking of this like clustering trend on the graph, it actually gets better as you carry it out more steps. It just gets a lot harder to display because even with this, there's already a lot going on. If you carry it out another step or 2 more steps, it becomes, uninterpretable, the number of molecules you see here. And then there's the other way we can do it. So before we laid it out using the multi-way graph, and then we colored it using the chemical space network. But if we did it the other way around, we get something that looks like this. So this is actually the same sort of system as before, except it's been carried out 4 reaction steps. And it's been laid out using a chemical space network with a similarity threshold of 0.9, as it was before. But now we've colored it using what we've termed the genealogical distance between 2 molecules. So if I go back up to this multi-way reaction network, and I wanted to compute the genealogical distance between 2 molecules, say this one in the bottom right and this one in the bottom left, I'm going to consider the shortest possible path that goes against the sort of the directed edges in this graph. And this is actually like a token event graph layout of this. So between this molecule here and then this molecule in the bottom right, we consider that one step. The orange dot just shows that there is a reaction taking place. This is a more useful layout if you have like multiple products. As you can see, like 1 molecule goes into a reaction and 2 come out. But we only consider this a distance of 1. So if we want to go from the bottom right to the bottom left over here, we have to go up 1, up again, and then down 1, down again. So we consider that a genealogical distance of 4. And you can do similar things for other ones in the graph. If you have like a parent molecule and a child molecule, that's a distance of 1. And then between this top one and everything in the bottom row is a distance of 2. So returning to this graph, the way it's been labeled is for each cluster, we've chosen a center molecule. So, and then every other color in that cluster tells you the distance between that molecule and the center molecule. So for example, in this big cluster here, the center molecule was chosen to be this one, and center, the way it looks in the chemical space network is not necessarily indicative of how close it is genealogically to all the others. The layout within each cluster is sort of just determined based off a gravity embedding. So it doesn't give you quite as much information as you might hope for genealogically. But looking at the colors, we can see that most of the components have very like orange and yellow and green. It's mostly those colors. There are some with very high colors, or sort of like blue shifted colors. So like in this one, you see a red and a blue. So these are very far apart genealogically. How at least it seems that way. Again, we run into the same problem of if we carried out this reaction like another step, some of these branches would coalesce. We get the same thing here. If we had carried it out 5 or 6 steps and then formed those connections, these two may actually only be 1 or 2 steps apart in the multi-way graph. Um, and then going through an example, so the monoterpene biosynthesis, the same sort of thing we looked at before. You can, for going only 3 steps out, you get these sort of graphs we saw before with steep drop-offs in the size of the connected components. And then you can see this graph here for the chemical space network with this threshold of 9 and a nice diversity of sizes of these clusters. It's not like they're all in groups of 2 or 3. And you get this graph that we saw previously. And here's what the chemical space network looks like if you only carry it out 3 steps. And as you can see, in these larger ones, you do get sort of like blue shifted. You get large distances genealogically, but then looking at the bigger one, we notice that most of these became smaller distances. Right. So if you carry them out further, you'll actually get smaller distances overall and better sort of clustering of the colors on the multi-way graph.
So it's like filling in the gaps between the different topologies, I guess. You kind of get more of a full picture.
Exactly, exactly. And then, yeah, so some future work, things that I was interested in looking at is branchial analysis is something I wanted to try. The problem is it doesn't currently work, as far as I'm aware, with directed cycles. And that can happen in these graphs. For example, we have in here, we do actually have a cycle. So if we go from this molecule here, there's a reaction here that takes you back to this one, and this one can go forwards into this. And so because we have a cycle like this, we can't do any branchial analysis directly on it using the current implementation in Wolfram. So trying to resolve that in some way
So
in order
what do you
to...
mean by branchial analysis exactly?
So branchial analysis, let's see. Yeah, so the way it works, as I understand it, you form, if you have a multi-way graph that doesn't have these directed cycles,
Mm-hmm.
you have like one starting node, then you have a clear progression of like how many sort of generations does it take to get to a particular molecule, or the minimum number of generations. And then you can talk about, okay, out of the ones in this generation, which ones share a parent? And so you kind of see here, right, in this graph here, the group on the left, they all seem to be it seems to be just one like complete graph. And so they all seem to share a parent node. And then there's some on the edges that only share a parent with like half of the ones in here. And so if you had a much larger set of reactions, but maybe they were more restrictive. So for example, if you specified, oh, this reaction can only occur if the molecule has like a ring of this size, or if it's, this specific sequence of elements uh, in the like chain connecting to a particular functional group, then you'd get a more sparse graph, and you would also be able to have, there would be less connections here, and you could make better inferences about the genealogies of the molecules based on just which parent nodes they have.
And so the branchial analysis effectively requires either a directed acyclic graph or a tree structure to work.
Yeah, specifically because of that, it doesn't work on at least the reaction networks I was looking into. Um, I suppose there are ways you could, modify the reaction network afterwards to sort of try and get rid of that and sort of say, okay, if you've already generated a molecule, don't worry about, or like don't put it back in the multi-way graph and then look at
Mm-hmm.
that. But that seems um, it would take away information from the structure that you might want to preserve. And so it'd probably be better to just modify or expand upon how branchial analysis works to try and account for that. And then
Yeah, so I
also
guess,
look,
yeah, sorry. I guess that would mean like, oh, you want to concentrate on one molecule and spread a tree in the reaction network from there. and look, or a dag from there, and look at the distances from that particular molecule as a root.
Yes. Yeah. And then there are certain problems with this, as I mentioned before. So even when you're displaying it visually, trying to go like 4 or 5 steps out with certain sets of reactions gets unmanageable because you get hundreds or thousands of different molecules. And even if you're not trying to visualize it, dealing with, the various places a reaction can occur on thousands of different molecules becomes very computationally expensive. So there's certain things you can do to account for this depending upon what your ultimate goal is. One of the things that I tried initially was if I wanted to look at polymerization reactions, so you have some kind of sugar molecule, for example, and you want to look at the polymerization of that. And so what you can do is you can say, okay, I'm going to have like a trimer. So you have 3 of the sugar molecules next to each other, and you want to add one on to make it a tetramer. So you allow for every possible tetramer from your starting trimer, but then you say, okay, I only want to keep the ones with certain properties, right? So that may cut it down from, depending upon where your 4th one can attach, it may cut it down from 100 possible tetramers down to 20, depending upon, the specifics you want with your ultimate setup. And then from there, maybe you go from 20 to 400, but then you cut it down to only 100. And so while it still is going to grow fairly fast, you do get to cut down on it. So you can go 2 or 3 more reaction steps out. And so just integrating this into the functionality of the multi-way network for the multi-way chemical reaction network and using it with the chemical space networks would be very beneficial for allowing these reactions to be carried out a few more steps. And then there's just some other stuff in this. This is part of a community post I made. There's just some other things about things that can be done in the future, looking at like other similarity metrics. So this was all done with topological similarity. You could look at things like which functional groups are present, and then the specific numbers of those. And there's a lot of other similarity metrics you could define and gain useful information from these networks from. And this was done largely with the help of Robert Nakbar over the course of the Wolfram Summer School 2024. So I'd like to thank him. And there are also references, and you can read through this in more detail and get the code for yourself off of my community post. Yeah. So
Excellent. Okay, so looking at the multi-way plot, the multi-way graph.
yeah, do you
So we
want the
have like
one?
single edges, right? So it's like in a chemical reaction, like there are multiple molecules coming together and doing something, and they change. Um, so like either like in a polymer situation, you have like this thing, and you have something coming in and attaching to it. So it's like in that situation, you have 2 inputs and then like 1 output. Or in the case of uh, you know, arbitrarily, you could have any number of inputs, and then an event happens, and you have like some number of outputs as molecules. Have you? Is there any interest in or utility in looking at all the inputs and outputs to these events?
Yeah, so if you're talking about, I suppose to clarify, the way that I've been working with it is you have sort of the molecules you've generated so far, and then there's sort of this like background of other molecules.
Mm-hmm.
And in the graphs that I've shown, you only show the molecules that you've already generated going into the next ones, and then you
Mm-hmm.
just sort of use the stuff in the background without talking about what those molecules actually are. Um, if you wanted to say, okay, I have 2 to 3 molecules that I know of, and they're going to come together and form, one or two products, you would probably use this token event graph. Because while it's not shown here, what it allows you to do is you could have multiple molecules entering this node here, this event node,
Hmm.
and then, it could produce two different outputs. You could have multiple arrows coming from this one event node. So there would be some use in that, especially if you wanted to look at like catalytic cycles, for example, right? You could have, A and B come together to form C and D, and then D splits to form E and F, and then F and C combine to give you A again, right? And then you have a cycle there that gives you A. You also use some other molecules there, and you add some product molecules that didn't get used up in that reaction. They just got formed. So you'd probably want to use a token event graph like this to give you a better illustration of which molecules are coming together for which reactions and how exactly do you go from molecule A plus some other stuff and get back to molecule A.
Right, right. Um, that's a good point. So on one side, the thinking about it, you can use hypergraphs where each hyper edge is directed in the sense that you have an input side with a bunch of nodes coming into the edge, and then an output side, a bunch of nodes going out to the edge. And but then that could be remodeled as a graph with these two types of nodes, and that basically one nodes being the molecules, and then the other nodes being the events. So
Yeah,
exactly
exactly.
like you described here, you have a token event graph
Yeah,
to keep
exactly. So
track of
yeah,
the
either a
hierarchy.
hypergraph or a bipartite graph where you have tokens or molecules and events. Yep.
Very cool. And yeah, so the autocatalytic cycles are very interesting to think about, and that's kind of one of the research topics that we're investigating um, in some projects currently. And yeah, like you say, you have this ring, and then you have like other molecules getting producted out of that, which don't really contribute back into the ring, which is kind of our side effects. And you might have also, random input things that kind of uh, feed into the cycle but aren't part of the cycle, like help it along maybe.
Hmm.
And just thinking out loud, like, would there be like two cycles that are kind of like adjacent to each other sometimes?
So adjacent to each other, how? Like a molecule that's like formed from one that doesn't get used up, like gets used up by a different cycle.
Yeah. Or I guess, there would be molecules which participate in multiple cycles in general.
Yeah, yeah, that definitely happens in nature. I'm not like super familiar with uh an example of one, but I'm sure that that definitely does happen. Um, Molecules are very versatile, so I'm sure there's some that are involved in dozens of different autocatalytic cycles, and it would be interesting to see like um, sort of a large graph that connects like a bunch of these cycles together.
Yeah, that's the thing that makes this difficult, as you mentioned. The versatility of the molecules really does add to the complexity of these and the quantity of these graphs, these chemical space networks. Just thinking about, I don't know, fatty acid chains or sugar polymers, because you could just, especially with amino acids, making proteins, because there was just like this chain of things that you can just keep adding on to, and um, you get very long molecules such as DNA can be like arbitrary. It's like, oh, what's the space of all the things you can make out of Lego bricks? it's infinite.
Very large,
Hmm.
very unwieldy.
Yeah.
Yeah, very cool stuff. Basically, just wanted to say it's great to see work on chemical networks here. We always have our interface with more the mathematics and the computation, but obviously, as many of us know, the chemical application of this rewriting business is super powerful. So, yeah, congrats on the project. It looks amazing.
Thank you.
Yeah. And yeah, thank you for excellent presentation.
Thank you for giving me the opportunity. It's quite enjoyable. Quite a nice break from the monotony of life.
Mhm.
All right. If there's no further questions or comments, I think I will be going.
All right.
So
Yeah.
thank you all.
We'll wrap up. Thank you again, Nicholas.
All right. Yep. Have
Thank
a
you,
good one.
everyone.